You told me that it stands for deoxyribonucleic, long word, acid. Everybody pretty much agreed with that. What else can you tell me about it? We said that what type of macromolecule is it? It's a nucleic acid, which means that its monomer is nucleotides. And what are the 3 parts of a nucleotide? You have, tell me the 3 parts to a nucleotide. You have a 5-carbon sugar, which we said we can call a pentose sugar. You have a phosphate group, right? And then we had the nitrogen bases. Bless you. Everybody remembers that part, right? That was pretty much what most groups had that up there. In this case, what's our 5-carbon sugar for DNA? What's its name? Pentose, it describes it, tells me that it has 5 carbons. What's the name of the sugar that's in DNA? What's the name of DNA? Deoxyribose. So the name of the sugar in DNA is deoxyribose. In RNA, it's just ribose. So deoxyribose is the specific name of it. The phosphate group, remember the phosphate is that PO4 3- group. It's one of those functional groups. And then nitrogenous bases, and pretty much everybody had those listed out. What were my nitrogen bases in DNA? We had A, which was adenine, T, which was thymine, G, which is guanine. I'm going to put the C. I'll try to slide it up. And the C is cytosine. And then most of you guys had on there A goes to T and G goes to C, which tells me that they're stuck together. Because what was that main structure that you said? How did you describe it? A double helix. So you told me that the structure for DNA was a double helix. So the double helix, if I'm not very good at drawing, but I'd have one strand twisted like this, kind of, and then the other strand would be like, it's kind of twisty. And if I were to pull them out and make it look like a ladder, I'd have 2 strands, and then I'd have the things in the middle, like that, right? So that's where it's like the double helix, like right there. The sides of it, if you were to kind of look at it like a ladder, the sides of the double helix are made of a bond. Do you remember what we called it? Probably not. It's called phosphodiester. That phosphodiester bond, and that's going to be between the sugar of one nucleotide and the phosphate of another one. And that's how they're held together on the sides. But what about in the middle? In the middle here, so if this is my sugar and my phosphates are on the sides, in the middle I've got my nitrogen bases. So here we've got your nitrogen bases. What holds them together? What type of bond? Not nitrogen bonds. They're hydrogen bonds. What can you tell me about hydrogen bonds? They're easy to break. They're relatively weak. Why do you think that would be a good thing? When we go to make a copy of our DNA, perfect, we can break it open and it doesn't take a lot of energy. So the sides are made of the sugar and the phosphate, and they're going to be held together with those really strong phosphodiester bonds. The center, those nitrogenous bases, are going to be held together with those relatively weak hydrogen bonds. If we were to look at your helix, most DNA I can tell you that most of the times DNA has a right-hand twist. And I'll show you what that means. So most DNA is going to have a right-hand twist. Some DNA has a left-hand twist. And we said we call that southpaw or Z-DNA because of that twist. Well, what's going to happen where the right and the left twist come together? No, it makes it weak. Just like with that piece of metal that Tony's over there bending, you end up with what they call hot spots. And it's an area of sort of like a weakness, if you think of it like it's a piece of metal where, you know, you're twisting on two opposite ways. And what we find in here is this is going to be an area where you usually get a concentration of mutations. Hot spots, an area of weakness. Why could that be an issue? Well, nobody wants mutations, right? That's generally bad. Mutation is where you have a change in the DNA sequence. If we have a change in the DNA sequence, you're going to have a problem. Even the word mutation sounds like mutant, which is a bad thing in general, right? In general, but not always. We talked about, you guys all knew what your DNA nucleotide was. Just in case you don't remember, you've got your phosphate group over here. Then you've got your 5-carbon sugar, which kind of looks like a house to me. I'm, again, not the best artist. And then I'm going to have a nitrogen base. What type of nitrogen base is this one? What type? We had two, you gave me, huh? Well, no. Your nitrogen bases. Right? You had your adenine, thymine, guanine, and cytosine. How can I tell them apart? What were the two broad classes of bases we had? Perfect. These guys can either be purines or pyrimidines. What was the difference? Purines were double ring structures. Pyrimidines were single ring structures. We said that your purines would be like pure silver, cost double. Ag is this one. So adenine is a purine. Guanine is a purine. It's a double ring structure, so it would look something like this. How about this one over here? Is that a double ring? No, it's a single ring. That's your pyrimidines. We said the pyrimidine would be your cytosine, your uracil, pyrimidine, or your thymine. So this one over here is going to be a pyrimidine because it's a single ring. This is all going to be, you remember, this is all review. The only thing that's might be new is now we have to pay a little bit more attention to our sugar. We have a carbon here. Every place that there's a carbon here, a carbon here, a carbon here, and a carbon here. How many carbons is that? 5. That's what makes it our pentose sugar. Well, why should you care about the carbons? Well, when we start looking at DNA, we have to give it some sort of directionality. And a long time ago, there's some scientists who decided that the way in which we do that is to number our carbons. So your carbons are always going to be numbered going in a clockwise position from that oxygen. So this is 1. This is 2. This one's 3. This one's 4. And this one's 5. And they usually have like a little prime after it, like a little math thingy. So you have the 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And we're going to start talking about our DNA strands based on what is at the top. It can either be the 3 prime end or the 5 prime end. And so when they say that, they're talking about which carbon is on that DNA strand is going to be facing towards the top. So what's new that we just saw? The number of bonds. So from A to T, you said you had 2 hydrogen bonds. From G to C, we had 3 hydrogen bonds. Which one's going to be easier? A to T. They also told you about this idea of complementary base pairing. So complementary base pairing. We can also refer to it after the dude who found it. It's called Chargaff's rule. And this is who told us that A is always going to bind to T and G is always going to bind to C. And the example that the dude did on the little video was that if I've got a 5 prime A, T, C, A, T, 3 prime, I should be able to figure out what the other strand of DNA looks like, right? So how would the other strand read? Well, what would it start with? Which carbon? The 3 prime, because remember they run opposite each other. So it's going to be 3, and then what's going to line up with the A? And then and then 5 prime, right? So that would be your complementary base strand. So if you know one strand of DNA, you can figure out what the other strand of DNA means or has. It also means that I could give you a question that says a DNA strand is 20% thymine, how much cytosine? How would you solve that? You would say, I'm sorry, I couldn't hear you, Katie, what?
20% would be A.
So if I have 20% T, you know that I would have 20% A, because A and T always go together.
And then there would be 60%.
There's 60% left, which means that it's got to be divided by
2.
just those 2 bases, so it would be 30% C. All right.